mechanism, and recycling of the Q to the QH2, the ubiquinone to the ubiquinol, and then back again has the net effect of also pumping protons across the membrane. So now these electrons are transferred to cytochrome Bf complex, and then to plastocyanin, which is a soluble protein in the lumen. Okay, so this is similar to cytochrome C, and now it's passed to photosystem 1. Okay, so we started photosystem 2, then we go to photosystem 1. Photosystem 1 has its own chlorophyll, so called P700, also named for the wavelength of maximum absorption, or the absorption spectrum. It absorbs a photon of light. The linear direction of the electrons is to go into ferredoxin. I actually drew ferredoxin as integral into the membrane. It should kind of be floating on the stromal side like this. So it transfers those electrons to ferredoxin, and then to ferredoxin NADP reductase, where it reduces NADP to NADPH. But recall I said that because you have now absorbed a second photon of light, the E naught prime for this electron is now lower than it was before, and therefore it's able to actually pass that electron back to the Q pool. A low E naught prime means that it's really good at donating electrons, okay? And then by pumping it back to the Q pool, you can then pass it again through cytochrome Bf and kind of rinse and repeat. And what this enables you to do is to make a ton of ATP, capturing energy from the sun, but not gated by the availability of terminal electron acceptors, okay? So the availability of NADP is going to gate or limit the amount that you can run this unless you can do this cyclic electron flow. And of course now, because we have protons that are present in the lumen, we can run them back through a chloroplast, ATP synthase, and generate ATP in the stroma. All right, are there any questions about that? Okay, so now we're going to briefly review the Calvin cycle and what we talked about on Wednesday. So recall I said the Calvin cycle starts with ribulose 1,5-bisphosphate. Because it is a cycle, okay, you have to regenerate that at the end. It is the definition of a cycle. So this ribulose 1,5-bisphosphate is the acceptor for a carbon dioxide molecule, which gets added in a process called fixation. Okay, this is catalyzed by an enzyme called Rubisco. Recall I said Rubisco is this really, really tiny enzyme. It's really, really slow, and also it evolved before the oxygen atmosphere, and therefore it's just as good, essentially, as capturing oxygen as it is carbon dioxide, which competes for this reaction and generates toxicity, including these byproducts. So in this fixation reaction, we generate 3-phosphoglycerate, actually 2 3-phosphoglycerates. This is a 5-carbon sugar plus 1 carbon to make 2 3-carbon sugars equals 6. We're going to today. The 3-phosphoglycerate can then feed into a process that looks like gluconeogenesis. So this is the process called reduction. So remember, 3-phosphoglycerate is above phosphoenolpyruvate, and so now we can shunt it into gluconeogenesis in a process that's called reduction. And what this gives you ultimately is fructose phosphate, fructose 6-phosphate, and then that can be isomerized into glucose 6-phosphate and then used to make starch, cellulose, etc. through a phosphogluconeogenesis reaction. Now, fructose 6-phosphate can be regenerated or turned back into ribulose 1,5-bisphosphate through a process called regeneration. And that's what we're going to talk about today. Now, regeneration is actually really complicated for a couple of reasons that you may have already noticed. So the first thing is that you're adding 1 carbon here, and I'm essentially telling you that you're taking 6 carbons out. Obviously, that cannot be true. So in fact, the numbers are that 6 carbons come in, this thing turns a bunch of times, 1 glucose comes out, and then the rest of those carbon molecules are kind of scrambled around to generate ribulose 1,5-bisphosphate. The other thing that might be a little bit confusing is that this is a 6-carbon unit, and this is a 5-carbon unit, and it's actually not completely straightforward to turn that 6-carbon unit into a 5-carbon unit, and so that's one of the other things that the regeneration process does. Okay, so let's kind of summarize this here, sort of summarize the problem, and then we're going to move on to nature's solution. All right, so we talked about 1 and 2 on Wednesday. We talked about fixation, and we talked about reduction. So now we're going to talk about number 3, which is regeneration. So recall I said this is a cycle, so regeneration is a heart of a cycle. It means that you generate the thing that accepts, in this case, carbon dioxide, so you can run it many, many times. And so essentially, what the Calvin cycle does is it to take 6 CO2 molecules, okay? This is something that's a 1 carbon unit, and turns it into 1 glucose, okay? That glucose is a 6 carbon aldohexose. And the reason I'm bringing this terminology up is that as we move through today's lecture, you're going to hear me talk a lot about ketoses and aldoses, and I want to remind you very quickly what they are. Hexose refers to the number of carbons, it's hex, and ose, which means that it's a sugar, and the aldose means that it has an aldehyde. A ketose has a ketone, and you will see it as I draw up these sugar molecules later in the lecture. Okay, so the Calvin cycle essentially starts with a 5 sugar and adds 1 carbon to it, and then gives you a 6 carbon aldohexose and regenerates a 5 carbon sugar. But how do you actually do this? This is a bit of a puzzle, and so I'm going to show you one of the ways that nature has solved this puzzle. So essentially, I'm going to frame it again this way. You have 6 5 carbon sugars coming in. Why do I need 6 5 carbon sugars? It's because we're accepting 6 CO2s. Okay, so each CO2 needs its own sugar acceptor. So this is going in. Okay, it's going into sort of this Calvin cycle box. And then what's going to come out is 6 5 carbons. Oops. This has to be true because it's a cycle. We have to regenerate the thing that went in. The other thing that goes in is 6 by 1 carbon units. This is our carbon dioxide, so this is also in. And what comes out is 1 by 6 carbon. Okay, so this is 6 by 1, this is 1 by 6. Ultimately, this is glucose, or really any other hexose, aldohexose. Okay, so the way that this actually works is you get 30 carbons from this, you get 6 carbons from this, and that gives you 36 carbons total to work with. And so what you have to do is now mix and match these different molecules in a way, in a polymer that ultimately regenerates these things right here. Now the thing that I want to really kind of emphasize right now is that there are actual rules to this process. Why are there rules to this process? It's because this process is catalyzed by enzymes. And so anything that's catalyzed by enzymes has to follow the rules by which those enzymes, or the rules of the reactions that those enzymes catalyze. And so the rules for this reaction are as follows. So number 1, or A, I guess, is that there's only one carbon transfer, and that's the one from Rubisco. Okay, so that's actually outside of this regeneration phase. There's only one place where 1 carbon is added or removed. Number 2, or B, is that there are only 2 carbon and 3 carbon swaps. Carbon swaps. These swaps themselves have further rules. They have more things that you need to keep in mind. The first is that it is always a ketose donor. And that will become really clear why that has to be the case when we go through the reaction mechanism. So it's always a ketose donor. And D is that it is always an aldose acceptor. Okay, so this is the thing that loses, -2 or -3 carbons, and this is the thing that gains, -2 or +2 or +3 carbons. Okay, so these are the rules that govern the enzymes that are going to kind of do this carbon scrambling that enables us to do the regeneration phase of the Calvin cycle and make ribulose 1,5-bisphosphate back. Okay, so now I want to go over an overview of kind of how this works in a very general way. And then we're going to go further into kind of the nitty-gritty details of this process. And it's diagrammed here on the board. It's also uploaded to Canvas. Okay, so we're starting here with 6 5-carbon sugars, that's 30 carbons total. And this is ribulose 1,5-bisphosphate. This is our CO2 acceptor. When we add CO2, 6 CO2 to the ribulose 1,5-bisphosphate, what we end up with is 12 3-carbon units, okay? 12 times 3 is 36, 6 times 5 is 30, plus 6 carbons coming in as CO2 gives us 36. And this is 3-phosphoglycerate. these 12 3-carbon units actually have slightly different fates in the Calvin cycle. And this is where it kind of gets a little bit scribbly and squiggly and complicated, okay? So the first thing that happens is that 10 of these, okay, 5 by 5, get combined together, again, to form 5 6-carbon units, all right? Now you can take one of those out as glucose, which is great. That's actually the point of the Calvin cycle, which leaves us with 4 6-carbon units, which are split themselves, into, sorry, let me just edit, where goes up, this goes down. Okay, I just want to edit because this line should come over here. All right, so now we have 4 6-carbon sugars right here, which are split into 2 groups of 2, which themselves have slightly different fates, okay? Now this is where some new chemistry comes in that you probably aren't all that familiar with. And the chemistry we're going to talk about in more detail in a moment, but essentially all it does is it swaps carbons between 2 sugar molecules, okay? So all we're doing here are swaps. This essentially is a condensation or carboxylation. This is a combination of taking 3 and turning it into a 6-carbon unit. And now we're just going to do a bunch of swaps that's going to enable us to regenerate our 5-carbon units that enable us to run this thing, run the Calvin cycle as a true cycle. Okay, so 2 of these 6-carbon units are then swapped with the remaining 2 3-carbon units that were generated from the Rubisco reaction. What happens here, as you can see, the donor is a 6-carbon unit, the product is a 4-carbon unit. So clearly this is a 2-carbon swap. Recall, down here, I said that the allowable transactions are 2 and 3-carbon swaps. So this one here is a 2-carbon swap. Now we have 2 3-carbon sugars, they take on the 2 carbons from the 6-carbon unit to regenerate 5 carbons. So 3 carbons plus 2 carbons equals 5 carbons. Now we're cooking with gas because those 5 carbons can go back into the Ribulus 1,5-bisphosphate pool to run the cycle. Now we have to take care of the rest of the molecules. We have 2 remaining 6-carbon units, 6-carbon sugars. What we do is then we take the output of this reaction, which is 2 4-carbon units, and swap it with these 2 6-carbon units. Now this is a 3-carbon transaction, which should become immediately clear to you because this is 6 carbons and this is 3 carbons, okay? So this is a 3-carbon swap. Those 3 carbons move from this molecule to this molecule, making a 7-carbon sugar, which I think is something we probably haven't come across before in 705. So now we have a 7-carbon sugar, 2 7-carbon sugars, and 2 3-carbon sugars, and we can do 1 additional swap. It should be immediately clear that this is a 2-carbon swap because 3 plus 2 gives you 5, 7 minus 2 gives you 5, and then these can all feed back into the Ribulus 1,5-bisphosphate pool using the activity of things like epimerases and isomerases. Okay. I want you to note that in every single case, because the ketose is the donor, the ketose becomes shorter, okay? The ketose becomes shorter, and the aldose becomes longer. So that follows as a consequence of the rules that are on the bottom part of this board. The other thing I want to point out here is that the number of carbons is always conserved. So there's only one place where you're adding carbons to the reaction, and that's right up here through Rubisco. Once you have your 36 carbons in the Calvin cycle, okay, the carbons are conserved. So 6 carbons come in on the top, on the top here, 6 carbons come out here, and then all of this is just switches and swaps to get you back to your 5 carbon pools that can start the cycle over again. The other thing I want to point out is that this is actually only one solution that will give you back 5 carbon sugars for the Calvin cycle. If you go to different textbooks, you may find different solutions, and the reason is because there's actually no magic to the order in which I presented it. Why is this? It's because cells are just bags of enzymes, okay? We draw the TCA cycle as a circle, but it's not a circle. Cells are just bags of enzymes. These enzymes collide with their substrates and make products, okay? And so what you have is essentially a bag of enzymes that contain NADPH and ATP, and ribulose one side this phosphate, and carbon dioxide, and all of these things. And as long as you have a ketose and aldose come together, you can perform these swapping reactions, okay? So this is one solution, but it is not the only solution. Okay. So, like I mentioned, the rules that I've drawn over here on this side of the board emerge from the chemistry of the enzymes that catalyze these reactions, okay? And there's actually two enzymes that we need to go over for this purpose. Okay, so the two enzymes are, let's see. One is called transketolase, and that's TK, abbreviated transketolase. Okay, this thing moves 2 carbon units. And we have another one that's called TA, or transaldolase. I think this is spelled properly. And this moves 3 carbon units. All right? We're going to go over this, but the mechanism by which these two enzymes operate differs, okay? They accept a ketose, they swap and generate an aldose, okay? The ketose gets shorter, the aldose gets longer, but the mechanism by which they do that will differ. Okay. So now I'm going to fill in the Calvin cycle on the board here step by step. Let's see. So we are going to start at this stage here. So we've just created glyceraldehyde 3-phosphate. Okay, glyceraldehyde 3-phosphate comes from pre-phosphoglycerate, which is generated in the fixation phase. And glyceraldehyde 3-phosphate itself is produced by essentially running a process that looks like gluconeogenesis in reverse. And so glyceraldehyde 3-phosphate is obviously a 3-carbon sugar unit. And so the first step, the first swap that we're going to talk about here, is taking these 3 carbon units and swapping it with fructose 6-phosphate here, which is a 6-carbon unit, to generate that 4-carbon, let's see, we'll put it here, to generate the 4-carbon unit here, and then the 5-carbon unit here. Okay, like I said, this is a 3-carbon transaction, and if it's a 3-carbon transaction, that means it's going to be catalyzed sorry, it's a 2-carbon transaction. Yeah, a 2-carbon transaction. So it's going to be catalyzed by transketolase. Okay. So if we take glyceraldehyde 3-phosphate and we run a transketolase reaction with fructose 6-phosphate, okay, we're going to make a 4-carbon sugar, and we're going to make another 5-carbon sugar. So the 5-carbon sugar that we're going to make is called xylulose 5-phosphate. I'm going to draw it here for you now. Okay, so this sugar is called xylulose 5-phosphate. 1, 2, 3, 4, 5 carbons. The other thing that transketolase produces, just want to make sure I'm trying to line this up as best I can, but I actually can't reach. Okay. The other thing that transketolase produces is something called erythrose 4-phosphate. So I'm going to draw that here. That is a 4-carbon sugar. this one's called erythrose 4-phosphate. The arrows have worked somewhat. Okay, so essentially what we've done is we've taken a 3-carbon sugar and a 6-carbon sugar, and we've moved 2 carbons from the 6-carbon sugar to the 3-carbon sugar, generating a 5-carbon sugar. Okay, that's 1, 2, 3, 4, 5. And the 6-carbon sugar is now 2 carbons shorter. Okay, so now we have a 4-carbon sugar. And this reaction is a transketolase reaction because it involves the movement of 2 carbon units. In this case, the carbon donor is the ketose, okay? The carbon acceptor is the aldose, and it regenerates a ketose and an aldose, okay? So regenerates a ketose and aldose, number of carbons are conserved, ketose is the donor, aldose is the acceptor, 2 carbons get moved around. We call this transketolase. I want you to note that xylulose 5-phosphate is an epimer of ribulose 1,5-bisphosphate. So what is an epimer? If you recall from the carbohydrates lecture, they're carbohydrates at different stereochemistry at one position. You can see there's one hydroxyl that's in an opposite orientation, and because they are epimers, they can be interconverted with ribulose 1,5-bisphosphate by something called an epimerase. So I'm going to add an epimerase onto the board right now. Okay, so this thing, which is a 5-carbon sugar, now can go back into the ribulose 1,5-bisphosphate pool by the action of an enzyme with an activity that we call epimerase activity, and that phosphate ATP. Okay, so where are we now? What we're going to do now is we're going to start with remaining 2 fructose 6-phosphates here. And this is, again, is our ketose. Okay, so if this is the ketose, that means it is the carbon donor. And now we're going to do a swapping reaction, and we're going to swap these carbons with carbons on erythrose 4-phosphate. So this is the donor here. This is the acceptor. We're moving 3 carbons this time, meaning if we start with a 3-carbon unit, we're going to end up with a 6-carbon unit. We're going to end up with a 3-carbon unit. And those 3 carbons are going to end up on erythrose 4-phosphate, generating that weird 7-carbon sugar that I demonstrated or I diagrammed up there on the board. So this is how we generate that 7-carbon sugar. So let's actually draw out the structure of these various molecules. So erythrose 4-phosphate comes down here and does a swapping reaction with this fructose 6-phosphate here, okay? I mentioned it's a 3-carbon swap, meaning it's a transaldolase reaction. If we're doing a 3-carbon swap using this one as the donor, okay, we're going to end up with a 3-carbon sugar. This is glyceraldehyde 3-phosphate. H, C, H, OH, CH2, OP. So this is also glyceraldehyde 3-phosphate, okay? And then what we're going to generate is the 7-carbon sugar, which we call cetoheptulose 7-phosphate, and the structure of that is here. I'm going to count my carbons, make sure I generate 1, 2, 3, 4, 5, 6, 7, and this thing is called, let's make sure I have room for it, cetoheptulose 7-phosphate, okay? Transaldolase reaction, 3-carbon unit is moved, starting with a ketose donor, generating an aldose that is 3 carbons shorter, and a ketose here that is 3 carbons longer. So now we have a 7-carbon sugar that can function as a ketose donor. Okay, the ketose is always the donor. And what we can do here is we can swap 2 carbons again through a transketolase reaction to make 2 5-carbon sugars if we combine it with glyceraldehyde 3-phosphate. So what we're going to do is combine it with glyceraldehyde 3-phosphate. So what we're going to do is we're going to run this through a transketolase reaction here. Okay, so this is a transketolase reaction. We're going to take glyceraldehyde 3-phosphate, which is this molecule, and also pass it through this swap. Okay? This is our aldehyde, meaning it is our acceptor. Okay, it's going to get longer by 2 carbons. So 2 plus 3 is 5. So that's going to give us a xylulose 5-phosphate. One, two, three, four, 5. Again, xylulose 5-phosphate. Okay, this will generate a ribose 5-phosphate. H1, 2. One, 2, 3, 4, 5. This is ribose 5-phosphate. Okay. Now we have 2 different flavors of 5-carbon sugars. So all we have to do in order to regenerate our ribulose 1,5-bisphosphate is pass these 2 molecules through epimerase reactions or isomerase reactions, which will move you between an aldose and ketose, and this will enable you to regenerate all of the ribulose 1,5-bisphosphate that you need. So these are both going to come here. Okay, so this one is an epimerase reaction. This one is an isomerase reaction. They both require ATP. So this diagram is available for you on Canvas, so you don't have to rely on like my very, very messy handwriting, but sort of in a nutshell, this is how the carbon swapping works, which enables you to regenerate ribulose 1,5-bisphosphate, which is the carbon dioxide acceptor, so you can continue to run the Calvin cycle. Okay. So now we're going to go through some of the mechanisms, or 2 reaction mechanisms, one for transketolase and one for transaldylates, okay? So the rules and the constraints for these transketolase and transaldylates reactions, as I mentioned earlier, are as follows, okay? They move different lengths, different carbon lengths. So transketolase moves 2 carbon units, and transaldylates, of course, moves 3 carbon units. And they do it always, I'm going to say this again and again and again, using the ketose as the donor and the aldose as the acceptor. Why is the ketose the donor and the aldose the acceptor? It's just about how the reactive groups are oriented and how the reaction mechanisms work, and sort of what reactive moieties these enzymes require in their active site in order to function. Okay? The other point I want to make here is that you can't rely on the names of these enzymes to understand which is the donor and which is the acceptor. And that's because they're actually not named, okay, for what is the donor and what's the acceptor. They both use ketoses as the donor. They just transfer. They do those swaps in a slightly different way. And it turns out that transaldylates is actually named likely because the reaction mechanism resembles an enzyme called aldolase, which you learned about when you learned about glycolysis. Okay, so there's two other things I want to mention before we go into the reaction mechanisms. Okay, the first is that transketolase requires a cofactor, and that cofactor is called TPP, or thymine pyrophosphate, okay? So transketolase requires TPP, transalkylase does an aldolase-like reaction. Okay, but it does not require TPP. Okay, so they actually have different requirements for cofactors, and this is because they catalyze this reaction in slightly different ways. All right, so let's start with the transketolase mechanism. I think I want to pull forward. So I'm going to start by drawing the active form of TPP, or a piece of the active form of TPP in the active site of this enzyme, okay? This has to have a negative charge. So recall what TPP does, okay? Like, what is the actual purpose of thymine pyrophosphate? So the purpose of this cofactor, before we actually go into the reaction mechanism, I want to go through what the purpose of this cofactor is, because I think it's important for understanding how the chemistry works. Okay, the purpose of this cofactor is to stabilize carbanions. So carbanions are carbon atoms that have an extra lone electron pair and therefore have a negative charge, and these things are wildly unstable, okay? And so what this thing does is actually stabilizes a carbanion, which it does by resonance. And this is very, very important if the chemistry that you're trying to catalyze has an intermediate generated that it does not have resonance stabilization. So that's the purpose or the utility of this TPP cofactor. You've seen TPP before, okay? Do you guys remember pyruvate dehydrogenase perhaps, or alpha-ketoglutarate dehydrogenase? Okay, PDH came in the mitochondria when you take pyruvate to make acetyl-CoA. Okay, alpha-ketoglutarate dehydrogenase is involved in the TCA cycle. Okay, both of them catalyze what's called oxidative alpha decarboxylation. TPP is really, really, really good at breaking carbon-carbon bonds. It turns out that TPP can also catalyze non-oxidative decarboxylation, or non-oxidative breaking of carbon-carbon bonds. In this case, it's not a decarboxylation reaction precisely, and that's the utility of this cofactor, and it's why it's being used by transketolase in its mechanism. So we're going to start here with our ketose donor. I'm going to endeavor to color the carbons in a way that kind of makes sense where things are coming and going. So here's our ketose donor. Right. All right, no H here. So this here is our ketose donor. And what we can do is we can use this lone pair of electrons to nucleophilically attack this ketone right here and pull a proton from solution. What this gives is the following intermediate. We've reduced this ketone to an alcohol. And now I'm going to draw what's on the other side of that bond. R. So now what can happen is a lone pair of this oxygen can come in and actually break this piece off. And what's going to leave is an aldose that's 2 carbons shorter. I'm going to draw a quick parallel with oxidative alpha decarboxylation here, okay? right now, what we're doing is we're doing chemistry at this position, at this alcohol, okay? If this was in pyruvate dehydrogenase, instead of this being a hydroxymethyl group, okay, it would be a carboxylate. And so if we broke the bond here and decarboxylated, then we would move on to the rest of the PDH reaction, which ultimately generates the 7-CoA. But in this case, we're actually doing chemistry on the other side. Again, this is a hydroxymethyl, it's not a carboxylate, and the result is that the aldose leaves. So now we're left with this. Okay. Okay, so now we're left with this intermediate. What we can do is we can now take a different aldose acceptor. So I'm going to denote the aldose acceptor using R prime. Okay, so instead of R, this one is R prime because it's our aldose acceptor. And so we're going to take this lone electron pair here. We're going to use the electrons in this double bond to form another carbon-carbon bond with this carbon here at this aldehyde. We pull a proton from solution. This gives us the following intermediate. Note, I'm going to put this as an R prime because it is a different aldose. And now we can regenerate the carbanion by breaking this bond right here. And what that does is it liberates back the active form of thymine pyrophosphate and gives you a ketose that's 2 carbons longer. Okay, so we accomplished a swap. This is an R group here. This is an R prime. All right, now let's go through the transaldolase mechanism. Okay, so the transaldolase mechanism. I mentioned earlier that this looks a lot like aldolase from glycolysis, and just like aldolase, it actually has an active site lysine that participates in a nucleophilic attack in order to sort of launch this reaction. So again, I'm going to try to color code the carbons so it's clear kind of where things are coming from. Pull a proton out here. This gives us a covalent intermediate in the enzyme. Now we're going to form our shift base and pick out a water molecule. So here we're going to use this lone pair to form the shift base. We're going to pick up this hydroxyl as a water molecule. we can have a Lewis base, remove this proton, which enables us to form the following chemistry. Okay, oops. And actually ultimately break this carbon-carbon bond. Because we're breaking a carbon-carbon bond, we're going to liberate the aldose. Okay, so this is an aldose minus 2 carbons. And it's an aldose that's derived from the ketose, and it's 2 carbons shorter than the ketose. Sometimes I find the minus and plus becomes nomenclature a little bit confusing. And we're left with this covalent intermediate on the enzyme. Oops. N, H. Now we can take it's actually minus 3 carbons. We'll put it here anyways, because essentially what you've done is you've clipped 3 carbons off. So now we have this covalent intermediate on the enzyme. We can now bring in another aldose acceptor. Right. Again, I'm going to denote this as R prime to distinguish this from the first R group that's present on the ketose that came in. So this is our aldose acceptor. And again, we use this lone pair to make the shift base to do chemistry, taking the electrons in this double bond, attacking, nucleophilically attacking this aldehyde and grabbing the proton. That gives us the following intermediate. Oops, this should be blue. Again, with our R prime, now we can bring in a water molecule. Right, that water molecule is going to come in here at this carbon-carbon double bond, or the carbon-nitrogen double bond, the shift base for the imine group. Forming this intermediate. And now, like this, we can take electrons from this hydroxyl to regenerate the ketone, because it's going to be a ketose that comes out and break this bond that regenerates the lysine. Okay, so the enzyme with that lysine has now been regenerated, and we end up with the following ketose that's 3 carbons longer. So this is a ketose that's 3 carbons longer. So the last thing I want to do is to actually draw out sort of what happens using kind of an exemplary ketose and aldose, so you can kind of understand what like the input is and what the output is and where the carbons go without really kind of focusing on all the movement of electrons, etc. So to do this, I'm going to basically give you a ketose and an aldose, but I want to really make the point that it doesn't have to be this ketose, it could really be any ketose and any aldose that participates in transketolase or transaldolase reactions. So this is our ketose. I'm going to color code the carbons like this. Okay, this happens to be fructose 6-phosphate, but it could be any ketose. Now if we combine it with an aldose, okay, this could be any aldose. It happens to be glyceraldehyde 3-phosphate. If we run the transketolase reaction, what we end up with is an aldose that's 2 carbons shorter, so it starts there. And then the remaining part of the aldose. Okay, this happens to be erythrose 4-phosphate. And then we generate a 5-carbon ketose. With carbons that are derived from this ketose donor and this aldose acceptor. So again, this could be any ketose. This happens to be hexagonase 5-phosphate. Now if instead of running the transketolase reaction, we run the transaldolase reaction, we're going to get something slightly different, okay? So transaldolase is moving a 3-carbon unit, this moved a 2-carbon unit. So if we remove a 3-carbon unit from this, what we're left with is a shorter aldose that's just the red business down here. So this is our shorter aldose. And a shorter aldose. In this case, it's a 3-carbon sugar because we started with a 1, 2, 3, 4, 5, 6-carbon sugar, and we removed 3 carbons from it. And then, now we have a longer ketose. So now we have a longer ketose. 1, 2, 3, 4, 5, 6 carbons here because we're taking a 3-carbon sugar and adding 3 carbons to it and making a 6-carbon sugar. Okay, so the numbers of carbons, if you add them up on the left and right side of the reaction, have to match. And this is a longer ketose that has 3 carbons more than the aldose donor did at the outset. So you can see how different enzymes will take the same molecules. Okay, here I've shown you F6P and G3P. 2 different enzymes can take the same 2 molecules and make different things. Okay, this is what I'm meaning when I say that, okay, these sets of reactions in the Calvin cycle can essentially scramble carbons around on these various sugars, as long as you follow the rules that I set out. Now, the last thing I want to mention is that it turns out that this is really useful for the Calvin cycle because you regenerate that 5-carbon CO2 acceptor, okay? But can you think of another reason why an organism might want to make 5 carbon sugars? I think I actually gave this lecture before spring break. It was the nucleic acid section, okay? So all organisms actually have a need for 5 carbon sugars because ribose and deoxyribose are part of our DNA, okay? And so obviously, organisms like humans don't have photocentrers, they don't do photosynthesis, but they still use transketolase and transalphalase to synthesize some of these really important 5 carbon sugars through the pentose phosphate pathway, which Professor Van der Heijden will tell you about after a very short break. So Tyler is going to put a polling question up.
All right. It's good to be back. First of all, congratulations. You guys did quite well on the exam. That makes me very happy. So good job. Let's do the polling question real quick. So, what do you guys think? Good. Yes. Okay. I think that's right. All right. Awesome. All right, so I want to pick up about how cells get ribose, right? You think about it, right? Cells have a lot of ribose in them because they have a lot of nucleic acids. Of course, there's a lot of RNA and DNA, but we've talked about a million other things, right? It's not just DNA and messenger RNA and sort of central dogma stuff. If you think about it, ribosomes, if you think way back to Professor Yaffe in the first half of the class, there's a ton of RNA as part of the ribosome. There's RNA components or nucleotides that are ADP, obviously, GTP, UTP, all these things we talked about, but also the cofactors, FAD, NAD, NADP, coenzyme A, tons of ribose in cells. In fact, it's been estimated that 50% of bacterial mass is nucleic acid. I'm not sure if that's true. That's not quite true in mammalian cells. It's about 20%. Someone in my lab did that a few years ago. But regardless, cells need a lot of ribose. And so where does ribose come from? Well, if you happen to do photosynthesis, you can run a different variation of the Calvin cycle, pull out ribose, cataplerosis. It's a cycle, right? Just like we talked about with the TCA cycle, you need anaplerosis, but nonetheless, you can pull ribose out that way. But we don't do photosynthesis, so we don't run the Calvin cycle, yet we still need a lot of ribose. So where does it come from? Well, a solution could have been, you might think, oh, I eat plants, so if I eat plants, I'll just get photosynthetic ribose. And it is true, you can salvage some nucleotides from your diet, okay? That's a source of ribose. But it actually turns out that most mammalian cells actually can't get just free ribose. We don't have an enzyme to actually capture free ribose. And so most organisms that don't do photosynthesis have to make it from other sugars. Okay, so they pull sugars in glycolysis like glucose and use that to generate 5 carbon sugars. And even beyond just a source of ribose, cells also need a source of NADPH. Right? That was another thing you could get from photosynthesis. But it turns out you also, not just plants, have to deal with oxidative stress, right? You have to keep your glutathione reduced. Did you guys talk about that last week? Okay, good. So you guys have to keep your glutathione reduced. You have to deal with all those reactive oxygen species. You have to do reductive biosynthesis, okay? If you're going to make fat, what we're going to talk about on Monday, you need a source of electrons for fat. Those come from NADPH. And so you also need, if you do photosynthesis, of course you can do photosynthesis to get NADPH, but you can't do photosynthesis, so you need a source of NADPH as well. And it turns out the way this is dealt with is what I want to spend time talking about now. So I want to remind you, so all organisms basically have these cofactors to deal with basically how they're going to do oxidation and reduction reactions. And so every single cell has an NADH to NAD plus ratio, an NAD pH to NAD B plus ratio, and these ratios are different in cells, all right? It turns out that this one is more oxidized, meaning that NAD is higher than NADH. This one is more reduced, meaning that NADPH is higher than NADP plus. Why is that? Well, because that allows you to do both oxidation reactions, glycolysis, the TCA cycle, whatever, while at the same time doing reduction reactions, right? Two different cofactor pools, you can carry out different things at the same time. Now, doing these reduction reactions or dealing with keeping your glutathione reduced, etc., you need to source something to charge up that NADPH, NADP ratio if you're a non-photosynthetic organism. And it turns out that the way that this happens, and probably how this happens, is that organisms evolved a way to repurpose what happened in photosynthesis as a way to do this for non-photosynthetic organisms. That is a pathway that effectively gives you NADPH, while at the same time gives you a source of ribose. Now, the pathway that is used to do this is something called the pentose phosphate pathway, which is often abbreviated the PPP, all right, pentose phosphate pathway, sometimes referred to as the pentose phosphate shunt. I'll explain why the word shunt comes in a second. But it turns out that this pentose phosphate pathway or pentose phosphate shunt is the major pathway that all cells use to get NADPH and ribose as an alternative to photosynthesis. Okay? So if you think about it, why would this ever evolve? Well, it evolved. Remember, we talked about this at the very, very beginning, right? Plants, they can do photosynthesis when the sun is up, but then nighttime comes. Turns out plants still have to deal with oxidative stress, and if they want to make stuff in the dark, right, they need a source of these things as well. And so they came up with a pathway to do this that was likely co-opted by us as heterotrophic organisms that eat plants to do it without photosynthesis. Now, the way that this pentose phosphate pathway works is it's conceptually akin to the Calvin cycle in reverse, right? So the Calvin cycle, what did we do? We used NADPH from photosynthesis as well as ribose to ultimately generate glucose, 6-carbon sugar. Well, if we start with a 6-carbon sugar and we run it in reverse, different pathway, of course, has to be a different pathway, as you know from the exam, then you can generate ribose and that generate NADPH, and that's effectively what the Calvin cycle is. And so what it is we're going to start with a 5-carbon sugar. All right, and what are we going to do? We're going to add a CO2 molecule to the Calvin cycle starts with a 5-carbon sugar, adds a CO2 molecule, reduces it with NADPH, and what we end up with is a 6-carbon sugar. Okay? Summarizing a lot of stuff right there, but effectively that's what the Calvin cycle does with all the details that you just heard about. All right? Well, what the pentose phosphate pathway is going to do is we're going to do the opposite. We're going to start with a 6-carbon sugar. All right, and we're going to effectively take away a CO2 and generate an NADPH and a 5-carbon sugar. Okay, so I'm going to just reverse this pathway in a lot of ways. Now, obviously, losing the CO2, we're going to then basically NADP to NADPH, here it's NADPH, NADP, whatever, the details we'll get to in a minute. But effectively, this is what we're doing, and if we do it that way, now we have a source of NADPH and 5-carbon sugars like ribose, which allows us to make nucleotides. All right, so why is this called a shunt? It's called a shunt because this is effectively a branch of glycolysis. You can think of it as an alternative way to do glycolysis that allows you to make these various intermediates. All right, so if we think about glycolysis, all right, so remember glycolysis, we start with glucose 6-phosphate, okay, that's the top of glycolysis. And turn that glucose 6-phosphate into the ketose, fructose 6-phosphate, okay, and also generate glyceraldehyde, phosphate. All right, effectively, go on, make pyruvate. Okay, this here is glycolysis. Removing lots and lots of details, but just for a couple key molecules there that are in it here that are relevant. Well, it turns out that if we do this conceptual reversal of the Calvin cycle, take our 6-carbon glucose 6-phosphate, we take off a CO2, and at the same time generate an NADPH, or NADPHs along the way. What do we get? Well, now we can get a 5-carbon sugar, ribose 5-phosphate being an example of a 5-carbon sugar, all right? And that solves what I need. However, I can also, there's no reason, once we have these transketolase and transaldolase reactions, there's no reason I can't use those again. There's nothing about photosynthesis that's special about that. These are just equilibrium reactions that are swapping around 2 and 3 carbon sugars. Well, I can easily just come up with a series of reactions that do these swaps that you just heard about between 5 carbon sugars here to make some 6-carbon and 3-carbon sugars, including things like fructose 6-phosphate and glyceraldehyde 3-phosphate that can then re-enter glycolysis. And so this direction is sometimes referred to as the oxidative pentose phosphate pathway. Why? Because I'm oxidizing my glucose, something else gets reduced, that makes NADPH, okay? And then I can get a 5-carbon sugar. Whereas this other pathway down here is referred to as the non-oxidative pentose phosphate pathway, which is effectively a bunch of transketolase and transaldolase reactions that allow me to interconvert 5-carbon sugars with 6-carbon and 3-carbon sugars, just like you heard about from Professor Ringel. And so really this is effectively 2 different pathways that are routes to interconverting things in glycolysis to making 5-carbon sugars. In other words, this oxidative pentose phosphate pathway is glucose 6-phosphate going to a 5-carbon sugar, ribose 5-phosphate, it could be a different 5-carbon sugar, plus CO2 plus NADPH. Okay, this is the OX DPP. All right. The alternative is this so-called non-oxidative DPP. Okay, what's the non-oxidative DPP? This is taking fructose 6-phosphate and glyceraldehyde 3-phosphate and basically using transketolase and transaldolase to interconvert it with ribose 5-phosphate. All right? No oxidation happening here, it's just this 2 and 3 carbon swaps by transketolase and transaldolase, non-oxidative PPP. The other is an oxidative pathway. Oxidation is happening. I'm oxidizing glucose 6-phosphate to lose the CO2. Electrons have to go somewhere. I can generate an NADPH and I get a 5-carbon sugar. You'll notice that both of these are pathways to get 5-carbon sugars. This is a shunt because you can imagine this is some flexibility here in how I can do this. All right, imagine the cell needs to make nucleotides, so it needs a bunch of ribose, and it needs NADPH. Well, great. I can start with my glucose 6-phosphate, run the oxidative pentose phosphate pathway, I get NADPH, and I can get 5-carbon sugars. All right, what if the cell just needs NADPH, it doesn't need ribose? Well, that's great. I can start with my glucose 6-phosphate, make 5-carbon sugars, get some NADPH from it, take those 5-carbon sugars, put them back in glycolysis. Now I can run them through glycolysis and get some ATP. All right? What if I don't need NADPH? I just need 5-carbon sugars to make ribose. Well, now forget the oxidative pentose phosphate pathway. I can start with 6-carbon sugars, fructose 6-phosphate and glyceraldehyde 3-phosphate, and make ribose 5-phosphate that way. So it's a shunt because it's an alternative route through glycolysis, but there's a lot of flexibility here, all right? Oxidative PPP, I mean, all reactions, I hope as you appreciate by now, are reversible, but under conditions that exist in cells, you're never really running the oxidative PPP back towards glucose 6-phosphate. NADPH, NADP's high, stuff like that. That's not happening. So this is pretty much unidirectional in cells, but the non-oxidative PPP can go back and forth. All right? Okay. Now, oftentimes, these pathways are discussed together, and it can get a little bit confusing because sometimes you're like, oh, it's a pentose phosphate pathway, it's the same thing. But don't think of it that way. Really think of these as 2 different pathways. One is a pathway that allows you to oxidize glucose 6-phosphate to ribose or 5-carbon sugars. The other is just interconversion of 5-carbon sugars and 6-carbon sugars. So they can work together as a shunt, but they can also work separately, just as I've talked about. Now, in the last 5 minutes that I have today, I'm going to quickly tell you about the oxidative pentose phosphate pathway. Okay, think of it as a different pathway. And then next time, I'll finish up and tell you about the non-oxidative pentose phosphate pathway, which will reinforce some of this confusing transketolase, transaldolase stuff, which is really how you interconvert those sugars. Okay, so what's the oxidative pentose phosphate pathway? Okay, so the oxidative pentose phosphate pathway, so I'm going to start with our old friend here, glucose 6-phosphate. All right, I'll draw it for you in the alpha position. So this is alpha glucose 6-phosphate. All right, and okay, so if I oxidize this 1 carbon of glucose 6-phosphate, just like we saw in epimerase reactions, okay, that hydride ion, in this case, can go to NADP. That's a source of NADPH. And what I'm left with is this lactone, okay, which is effectively the aldehyde that was at the 1 carbon is now an acid, but it's in ring form, okay? This molecule is called 6-phosphogluconolactone because it's glucose with a lactone in the ring. See this if I come along and just open up the ring, which is what happens next. Okay? So basically opening up the ring with water, a so-called lactonase reaction. Draw this now in the open chain form. Okay, so this is 6-phosphogluconate, right? And it also demonstrates how that what was the aldehyde on glucose is now an acid, right? So the first step did that. This is catalyzed by an enzyme called G6PD. I'm not going to draw it out for the sake of time. Glucose 6-phosphate dehydrogenase. Next thing I'm going to do is I'm going to oxidize this 3-carbon. All right, so if I oxidize this 3-carbon, okay, again, hydride ion comes along. This can go to another NADP plus, NADH. Okay, and what I'm left with is this intermediate. For the sake of time, I'm not going to draw the whole thing out. But if you look at this, this is a beta keto acid, okay? Acid is beta to this ketone group. We've seen now this a million times. This is favorable for decarboxylation, okay? Now I lose my CO2. What do I have now? Now I have this intermediate. And for the sake of time, I'm not going to draw this out. It's just the top part there of the molecule. What's this? This is an enol, okay? Enol likes to rearrange, and what I'm left with is a carbon sugar, which I hope you recognize is ribulose 5-phosphate. And once I have ribulose 5-phosphate, I can use isomerases, or I can use epimerases, and I can convert this to ribose 5-phosphate or xylulose 5-phosphate, which are great substrates for use in transketolase, transalpylase reactions that can give me back to 3 and 6 carbon sugars, which I'll show you next time. Okay? Perfect. Have a great weekend. It's good to be back. I'll resume my office hours today at 5, and I'll see you guys on Monday.